Hello everyone, my name is Juliana Virginio da Silva e I'm a doctorate student at Federal University of São Carlos and São Paulo in Brazil. I am currently developing my research projects at Embrapa Livestock Southeast, also located in São Carlos, São Paulo. I am grateful to present my results in this congress. The title of my presentations is Dietary Impacts on Rumen Metatranscriptome of Nelore Cattle. The rumen is a digestive structure in ruminant animals that convert low-quality food in high-quality proteins through the actions of microorganisms, including bacteria, archaea, and eukaryotes. Studying the ruminal microbiome has shown promise for understanding ruminal fermentation and improving animal proteins conversion. Advances in DNA and RNA sequence have enabled the study of the rumen microbiome through approaches such as metagenomics and metatranscriptomics. Traditionally, studies have utilized metagenomics to explore the genetic compositions of microbial communities in the rumen, revealing their taxonomic diversity and potential metabolic capacities. In turn, metatranscriptomics allows scientists to study the profile of activity genes expressions in ruminal microorganisms. So, metagenomics and metatranscriptomes have been applied to investigate alterations in the compositions and functionality of the ruminal microbiome. However, few studies have integrated these powerful omics to compare how different diets affect the ruminal microbiome of beef cattle. So, understand the lack of research in this area. Our studies aimed to understand more about how dietary changes affect the gene expressions profile in the rumen. For these reasons, we integrate metatranscriptome data with 447 previously constructed metagenomics assembled genomics (MAX). We focus on compare the expressions profile between two groups of 26 bulls that received distinct dietary treatments, conventional or byproducts. About the methodology following the work, the experiment was carried out in Embrapa Livestock Southeast. These 52 animals were submitted to two different diets. One group received a high-grain diet consist of corn silage, core grain, soybean meal, rumen protect fat, and urea. And the other received corn silage, citrus pulp, peanut meal, corn germ oil meal, and urea. We collect the ruminal content samples and follow the steps: RNA extractions, sequences through Illumina HiSeq, quality control, and pre-process of the reads. The next steps involved bioinformatic approaches and data analysis. First, use the Proca software to obtain the files containing the code regions and proteins of the mags. Later, used Cofan Koala to obtain the functional annotations of these mags. About data integration, use the Callisto tool to perform a mapping between the metatranscriptome data in the code regions of the mags. And using the RStudio software, we'll combine the Callisto output with the functional annotations of these mags, obtain a file with normalized expressions levels per mag and per sample. On the results obtained in this work, we'll note that about 960,000 code regions were obtained, and around 884,000 had hits against metatranscriptome. For the last, about 15,000 genes with no functions were differentially expressions between the diets, considering the Wilcoxon paired text. So, our research has demonstrated that dietary variations can significantly influence the functionality of the rumen microbiome. This affecting the expressions level of genes associated with amino acids, carbohydrate, energy, and lipid metabolisms. This finding underscored the importance of understanding how dietary factors can modulate gene expressions in bovine rumen. In other words, depending on the diet, different pathways are activated, for example, the degradation and biosynthesis of fatty acids. Another result obtained in this work indicates differences in expressions level between phyla. This indicates that diet can affect the metabolic activity of the ruminal microbiome, resulting in different gene expressions profile between phyla. In conclusion, our study underscored the intricate relationship between diet and the ruminal microbiome, revealing that different diets can lead to variations in gene expressions. Therefore, understanding the impact of diet can lead to better animal health and more efficient livestock production. Thanks for the opportunity. Comments and questions can be sent to email juvirginiosilva@gmail.com.